In this module of biotechnology, let us see the applications of biotechnology in the field of medicine. Now, when we say applications in medicine, there are many things. First, let us see genetically engineered insulin. Before we talk about the details of production of this genetically engineered insulin, let us first understand the basic structure of normal human insulin produced in our body. Insulin is produced in beta cells of islets of Langerhans, which is in pancreas. It is synthesized in the form of proinsulin or proenzyme. What is proinsulin? It is the inactive form of insulin. And the third point is the structure of insulin. Insulin consists of 3 polypeptide chains. A polypeptide chain, B polypeptide chain and C or linker polypeptide. So basically if we stretch the entire structure, it is going to look like this. This is A, then comes C, then B. So there are 3 polypeptides and this C chain or linker, the role of this C chain is to bring A and B closer so that between these sulfur containing amino acids A and B, a disulfide bond can be formed. And as soon as this disulfide bond is formed, then the role of this C, that is the linker polypeptide, is completed. So the C peptide is not found in the mature insulin and it is gradually removed or it will gradually disappear. It is released into blood during maturation and has no known biological activity. Now, scientists thought that what is the need of C when we need only A and B to synthesize regular insulin? A chain has 21 amino acids and B has 30 amino acids. When we say that insulin has 3 polypeptide chain, there are 3 genes responsible for this. That is, there is a gene responsible for A polypeptide, there is a gene responsible for this C polypeptide, and there is another gene which is responsible for B polypeptide. So what scientists have done is that they have isolated the gene for polypeptide A and gene for polypeptide B. Both these genes were first allowed to transcribe. When they transcribe, that is, when gene undergoes transcription, it produces hnRNA. This hnRNA has both coding and non-coding sequences. That is, coding sequences or exons and non-coding sequences or introns. Then this hnRNA undergoes splicing, capping and tailing to form mRNA. So, what scientists have done in this case is they isolated this gene and synthesized mRNA first. That is, RNA which has only coding sequences. Then using reverse transcriptase, this was converted into a DNA strand and this DNA strand is called as cDNA. Now, what is the need of doing all this? For this splicing and ligation, we need enzymes. And these enzymes are eukaryotic enzymes because insulin is a eukaryotic protein, whereas The host cell which we are using here is E. coli, that is a prokaryotic cell. So these prokaryotic cells do not have enzymes which is required for the process like splicing and ligation. So this cDNA which is produced are introduced into E. coli. When we say gene of a polypeptide, it is actually cDNA of a gene. This cDNA is added into a plasmid and this plasmid is introduced into E. coli. That is, this recombinant plasmid is introduced into E. coli. In the same way, it was performed for gene B also. They obtained cDNA for B gene. It was incorporated into the plasmid and this plasmid is introduced into E. coli. So, A gene was introduced into one E. coli and B gene into another E. coli. That means this E. coli will be producing only A polypeptide chain and this E. coli will be producing only B polypeptide chain. Then, after this, these two polypeptide chains were isolated from this E. coli and were kept together in conditions where disulfide bonds and thus insulin was synthesized. So, thus functional insulin is obtained by genetic engineering. This insulin was called humulin and the entire thing was done by a pharmaceutical company called Eli Lilly. And Eli Lilly introduced this humulin in the market on July 5th, 1983. I repeat the process. They isolated genes for A and B, obtained cDNA by this process to overcome the problem of producing eukaryotic protein in prokaryotic cell, and that cDNA was introduced into plasmid to obtain rDNA and rDNA was introduced into E. coli. So separate genes was introduced into different E. coli, that is A gene into one E. coli and B into another E. coli, and finally disulfide bonds are produced to form insulin. In 1998, Eli Lilly and Ranbaxy launched diabetic drug Humalog, an analog of human insulin, which is more expensive than other insulin products and is absorbed by blood within 10 to 15 minutes as against 30 to 60 minutes time frame of other regular insulin products. So, this is the first application that we have talked of in the field of medicine. We'll discuss other applications in the next video. Thank you.